Right, sounds good. So I'd like to start by thanking, the organizers, Ram, and, other organizers of bootcamp for the invitation to talk a little bit about microfluidics and mostly about the microfluidics work that we do in my lab, and welcome everyone to the bootcamp, and I hope you guys are enjoying the camp. Um, what I'm gonna do today is I've been asked to talk about microfluidics, but to make it more interesting, I thought maybe I would give a quick introduction, to go over the concept of microfluidics, and then, talk about the work that we do in my lab as an example of a recent trend in microfluidics. So that's actually my plan for today. And I would like this to be more of an interaction. So, feel free to stop me and ask questions, along the way. And I'm going to be covering a lot of different topics, involving different organs and tissues. And so some of them might be of interest to you, some of them might not be interest to you. But again, you feel free to stop me and ask questions anytime. Okay. Before we get started, any questions? Okay, good. So I'll try to finish by, by 1 o'clock. And so microfluidics actually, just to briefly introduce myself for those who don't know me, I'm an associate professor in bioengineering at Penn. And my lab at Penn is focused on developing organ-on-a-chip model systems for a variety of applications, including drug testing, environmental monitoring and screening, and so on. So I'll, show you some of those examples during this talk today. So I guess one of the first questions I have to say, I teach you a course on microfluidics, biomicrofluidics in the fall semester. And, when I start, this course, one of the first questions I ask to students is, so what comes to mind when you think about microfluidics, right? Like, so when you hear the word microfluidics, what are the representative images that you picture in your heads? And so when I ask this question, a lot of people, say, those microfabricated chips or point of care diagnostics and microfluidic point of care diagnostics or cell culture devices. These are common answers that I get every year. So a lot of people tend to think about these small scale devices, actually chips, used for a variety of applications. And so one could think maybe microfluidics is all about creating these chips, micro devices, but what's more important is, let me actually get this out of the way. So using these small-scale devices, channel systems, you basically gain the ability to, You gain the ability to control very, very small amounts of fluids in a highly controlled manner. Okay, so simply put, microfluidics is all about small plumbing or creating, smart plumbing system or small plumbing systems. So I'm showing here a couple of examples of, well-known microfluidic actuations or transport phenomena. So what I'm showing here is you can use this, well-designed T-junction, kind of geometry in a microfluidic device and along with two immiscible fluids. So in this case, the water is colored blue and this continuous phase is oil. So you can actually use this microfluidic design to form these microscopic droplets in a very high throughput fashion. You can generate thousands of droplets per second, which is surprising. And you can actually you can encapsulate, different chemicals or cells, biological, biochemical materials in these droplets and use these droplets as, individual chemical reaction chambers, right, to conduct biochemical experiments in a very high throughput fashion. Another example shown here in this movie is a multi-layered microfluidic device consisting of multiple layers. And so by using compressed air, you can, valve and pump fluid flows actually in microfluidic devices for analyzing biological materials. And so the example shown here, this technique is called multi-layer soft lithography, developed by Steve Quake's lab at Stanford, and it became the basis for starting this company actually called the Fluidigm, which sells gene sequencers and nucleic acid and analytical platforms. And another example, you can inject multiple types of liquids into a common channel and have these different liquids flowing right next to each other without any convective mixing, turbulent mixing. So using this technique called the multiple laminar stream technique or laminar flow patterning technique, you can develop very novel assays, for biomedical applications. Again, it'll be a stretch to cover all these different, techniques and technologies, microfluidic platforms in this short introduction. But just know that microfluidics is not about, it's far more than just developing these small-scale chips and devices. Using those, microfabricated structures and channels, you gain the ability to handle very, very small amounts of liquids precisely that would be almost impossible to handle using traditional laboratory techniques. Okay? Any questions about that here? Okay, so, just kind of, formalize this discussion, if you will. So, what I'm showing here is the definition of microfluidics given by the luminary in the field, is a pioneer of microfluidics as we define it today, George Whitesides. He's a chemistry professor at Harvard. He's the person actually with highest H index in chemistry in the field of chemistry as of today. So, according to his definition, so he gave out a definition in his Nature review published in more than 10 years ago that talked about the present and future of microfluidics. So he defines microfluidics as the science and technology of systems that process or manipulate very small amounts of fluids using channels with dimensions of tens to 100 micrometers. So that's basically what we just talked about. Okay, so again, there are so many interesting things we could talk about, with regards to microfluidics. But to kind of segue into my second, the second part of my talk. So, this actually graph shows the number of publications, and patents over the past. Well, actually, it ends in 2011, but microfluidics, actually, a lot of pioneering studies were done in the 90s, and then entering a new millennium, actually, it really took off, and then you can see this exponential increase in the number of research, papers and patents. And so, to talk about what's happening now, so I often use this paper. Again, this is now becoming an old review article, but the person shown in the photo is Dave Beebe, a biomedical engineering professor at the University of Wisconsin-Madison. And his group actually published a paper, review article in Nature, to talk about present and future role of microfluidics in biomedical research. And so he has actually a section in this paper entitled When Micro Tests. Micro Tests stands for Micro Total Analysis System. So this is actually used synonymously with lab on a chip, that many of you actually have heard about. So microtechnologies or microfluidics technologies are the only solution. So this is a very interesting section that talks about very unique opportunities, in biomedical research, that can be by microfluidics engineers. And there are three kind of areas of investigation he highlights in this article. The first one is diagnostics for low resource limited resource settings. And so this is a good example would be, using paper to develop paper microfluidic diagnostic chips for, limited resource settings and, for underdeveloped, settings. And so this is actually, this section actually talks about that. Another subsection is titled Rapidly Assaying Biofluids with Microfluidics. So you can imagine developing microfluidics-based, diagnostic tools for clinical use, right? And the third area that he highlights in the paper is modeling, physiological processes using microfluidics. So I've been fortunate enough to get involved in pioneering, this area of investigation in microfluidics. So what I'm going to do, again, my goal today is to give you a flavor of what's happening in microfluidics, today. Okay. So to achieve that goal, what I like to do is to talk about the work that's going on in my lab that actually falls under this category. So more physiologically relevant in vitro models, and microfluidics technology has played a crucial role in developing these more physiological in vitro model systems for biomedical research. Okay, so that's actually what I plan to focus on in the second or in the next section of my talk. So I have to say this is, very broad and, probably not such a good introduction to microfluidics, but I, you get the idea, right? So microfluidics is all about using these small scale channels to handle small amounts of fluids precisely, okay? So that's actually the key takeaway from the first section of my talk. Any questions about this? If you are, if you prefer, you can also use the chat window to, type in your questions, okay? So I'll be, watching this chat window as well during my talk. Any questions? Okay, all right. So let me just check the time here. Okay, 15. All right, so let me actually start this part of my talk. So I mentioned that my lab is working on developing organ chip, tissue chip model systems. So idea here is to, as some of you may know, what we do is we use microengineering and sometimes nanoengineering techniques to build these microchips. And this device, for example, shown here, has multiple channels and colored differently. What we do with using these channels devices is that we use them for culturing human cells. So whenever possible, we use primary human cells. Let me actually taken from human subjects, normal or diseased human subjects. We also started using iPS, derived cells or stem cell derived, specialized cells in these devices. But we also use engineering techniques to mimic very complex 3D structures and dynamic environment of human tissues and bodies in these systems, okay? And the idea is to use these systems to model these different aspects, very complex features in ways that have not been possible using traditional in vitro culture techniques like, Petri dish culture or 3D culture. So the idea is to use these systems as microengineered models of human tissues and organs. Obviously, we are not mimicking whole tissues or organs. We're actually focusing on most critical functional elements of these complex, systems in the body. So these systems are collectively known as human organs on a chip or human organs on chips. There are different ways to call these devices. Some people actually call them tissue chips. In government agencies like NIH actually, prefer this term, microphysiological systems. So these are all referring to the same, systems or same things, okay? So what I'd like to do today in this, kind of short talk is to give you a quick introduction to this technology and perhaps, help you better understand, what this organ on a chip technology allows, scientists and researchers do in biomedical research using, several examples of organ chip systems that we are developing in my lab at Penn. Okay, any questions so far? Okay, good. All right, so if you are new to this, so I'm assuming that many of you have heard about organ chip technology, but if you're completely new to this, one of the first questions you could ask is, how do we actually build these advanced engineering systems, right? How do we actually go about designing and creating organ and tissue chip systems? What are the design principles that engineers use to build these model systems? So the design principle that we use for organ chip technology is based on a reductionist approach. Okay, so oftentimes, so for this discussion, let's say you would have to have a target organ you like to mimic, right? So let's say we are interested in modeling the lung and developing a lung on a chip system. So then the first step in this design process is to closely examine the anatomy of the target organ. So you have to learn about the anatomy of the target organ, and any organ, virtually any organ in the body, can be broken down into the most critical elements called the functional units, okay? In the lung, those would be the alveolar air sacs or alveolar system shown in this, illustration. In the liver, for example, those would be, liver lobules or liver sinusoids. In the kidney, those would be, functional units would be nephrons. Okay, so we break the target organ down into, this functional element unit. Then once you identify the functional unit, the next step is to closely examine the functional unit to identify the key features, key structural features, key environmental features you would like to replicate in your device. So oftentimes we start this process by focusing on the structure of the functional unit. So what I'm showing here is this section showing the alveolar capillary interface between air and blood. So one of the most critical function of the lung is gas exchange, and to enable that, to make that happen, the alveolar air sacs are covered with blood capillaries. And so I'm showing the barrier between alveolar air sac and surrounding one of the blood capillaries. So as you can tell, there's a thin layer of specialized lung cells, the lung tissue. And on the other side, there's a thin layer of this specialized cells called endothelial cells, capillary vascular endothelial cells, lining the inner surface of this blood vessel. And there's a very thin interstitium between these two layers. So looking at the section, we would say, oh, so this is the key structural feature we want to mimic. So we want to mimic this three-layer structure consisting of lung tissue on one side, capillary tissue on the other side, is separated by a thin interstitium in the middle. So then once you identify the key structural features, then the next step is oftentimes, you can do different things, but one of the things that we do is to pay close attention to the environment these cells, the tissues are exposed to. So in the lung, as you can see in this animation, what's really interesting is that these alveolar tissues expand and contract, like balloons, as shown in this animation during respiration, during breathing, right? As we breathe in, air enters the lung, it travels all the way down to the alveolar air sacs, inflating these lungs. So this, kind of dynamic tissue deformation would be a very interesting and important environmental feature that we, would want to recreate in our device. And it's not shown very clearly in this animation, but there's also flow of blood through the vasculature, through this blood compartment. And the lung tissue is exposed to air. So these are all important environments, a cellular microenvironment that you want to recreate in your device, okay? So once you identify those structural features and organ-specific environment, the next step is to then go ahead and design a microfluidic system that would allow you to mimic those essential features. So this is kind of a quick rundown of the design process that microengineers use to build organ and tissue chip systems. So this is actually an example. So in this example, I'm showing this example. This is a study that I conducted as a postdoc a long time ago, but I think it's still a good example to talk about. So based on these features that we have identified, what you can do is you can design a system like this. This is a photo of the lung on a chip, breathing lung on a chip device. This is a cutaway view, and it's animation showing what's happening in this channel system. So there are a lot going on in this animation, and so I like to draw your attention to this middle compartment first. So this, there are two chambers, upper and lower chambers, separated by a thin porous flexible membrane made out of PDMS. It's a silicon elastomer. It's a silicon rubber, the most popular material that microengineers use in microfluidics for rapid prototyping purposes. So you can design a system and create these two chambers. On the upper side of the membrane, we can culture lung cells taken from human lungs. On the other side, culture capillary cells from human lung blood vessels. And so this is actually the three-layer structure we just talked about on the previous slide. And to mimic the environment, actually, so we flow media over the surface of capillary cells in the lower channel to mimic hemodynamic flow, blood flow. We also culture lung cells under air in the upper compartment to kind of mimic that environment in the lung. And to mimic this, tissue deformation during breathing, we came up with this design where we create these hollow chambers right next to the cell culture chambers. We apply cyclic vacuum to these channels. And because this material is soft and flexible, these vertical walls, separating these two compartments bow out like this, causing the stretching of the membrane as well as tissue layers attached to it. Okay, so using this platform, we can now, mimic the key, structural feature of the alveolar system as well as this dynamic environment of the alveoli in human lungs. Okay, any questions about this? Okay, so yes.
Do you also replicate the pressure and like the sort of pressures that a cell would feel in an actual lung?
So good question. So we do not in this particular study, in particular system, for a couple of reasons. But as you can imagine, in real lungs, what happens is, this compartment is pressurized, right? So this deformation in reality, it happens in a three-dimensional space. These three-dimensional balloon-like structures inflating and deflating. So one could actually come up with a design to mimic this realistic deformation in a system like this. But the reason why we shied away from it was because of imaging. So, these tissues deforming out of the plane actually would be very hard to capture and analyze. So that was the reason why we decided to go with this kind of a 2D, configuration. But to answer your question, we actually do not mimic pressure, and probably that could be one of the limitations. But these lung cells are sitting just at atmospheric pressure, and we flow air over the surface of cells, but we don't induce any pressure changes to mimic breathing. Does that answer your question?
Yes. Yeah.
Okay. All right. Okay, any other questions? Okay, so I was just saying, two important things to achieve are we mimic the structure, mimic the environment, but one of the reasons why many people are very excited about this organ chip technology is because of this, what I'm showing here. So using these organ chip devices, more sophisticated, more complex, organ chip in vitro model systems, we can now start thinking about mimicking more complex, organ level, not tissue or cell level responses, but organ level functions and responses that arise, from multiple tissues talking to each other. One of the common things that, engineers do in these systems is to culture more than one cell type. So in this long-on-a-chip example, epithelial cells are cultured with endothelial cells, right? By having these different cell types talk to each other, we can mimic more complex functions. They're kind of analogous to, emergent properties in complex systems consisting of multiple interacting components. So if you're not clear on this, and so let me actually, try to kind of, explain this using this example. So let's actually think about lung infection as an example of very complex, organ level responses that happens in the respiratory system. So let's say like we have, we're infected with bacteria, or, this is very relevant to what's going on now, right? So like the, virus infection, bacterial, in the lung, then one of the interesting, important things that these pathogenic insults do in the lung is to stimulate these alveolar lung tissues to produce and secrete pro-inflammatory cytokines, those chemicals that mediate pro-inflammatory responses in the body. Okay, so these pro-inflammatory cytokines and chemokines diffuse into the surrounding tissues, and when they reach the blood vessels in the surrounding capillaries, in the surrounding tissues, they actually activate these endothelial cells lining the inner surface of a capillary blood vessel. Okay, so what do I mean by activation? So when these endothelial cells are activated, they express adhesion molecules, surface receptors that are recognized by and engaged by immune cells circulating in the blood. So to kind of, explain this process of immune cell engagement and recruitment, I use this movie for engineers, So let's say we have these neutrophils circulating in the blood, and as you know, they don't do really anything, under normal circumstances. But when they see these, pro-inflammatory cytokines and activate endothelial cells, as you can see here, they stick to the activated endothelial cells. They squeeze themselves through capillary cells into the alveolar air sacs, and once inside the air sacs, they start crawling. This is not exactly how they crawl, but they crawl to the site of infection, and they start engulfing these bacterial cells to resolve infection. So this is actually one of the most complex organ level, not tissue level. So this process actually is a multi-step cascade involving multiple types of tissues, right? Like endothelial cells, immune cells, epithelial cells that secrete pro-inflammatory cytokines. So this is one of the most complex organ level responses that happens in human lungs and in animal lungs as well. So the question is, can this man-made system mimic this? So this is actually one of the questions we had in this study. And to address this question, we did an experiment where we got some human neutrophils. We fluorescently tagged them and put it into this lower vascular compartment. As you can see in this top-down movie of the vascular compartment, they actually just zip by without doing anything interesting. But when we put bacterial cells on the lung surface to mimic, pulmonary infection, take a look. So these neutrophils now stick, to the endothelial cells, to the lower side of the membrane, presumably because these endothelial cells are activated, expressing these surface receptors like adhesion molecules due to the stimulation by cytokines produced by infected epithelial cells. And the next movie shows the whole process. This is my favorite movie. So the neutrophil sticks, curls around. Cells are everywhere, but they're not labeled, okay? At some point, it starts wiggling its way through the capillary cells and one of the pentagonal pores on the membrane to get across the tissue layers from the vascular compartment to the upper compartment where the lung cells are. And you can see this guy go out of focus, and you can, that's because, it's moving in the vertical direction. And once in the lung compartment, these white cells, colored red in this movie, chase the bacteria and phagocytose them, engulf them, just like what happens in the living human lung, right? But the beauty of this is, not only can we mimic this, but also we can also visualize and, study this in real time at high resolution, which is one of the, compelling advantages of this organ chip systems over animal, models. Okay, any questions about this system? Yeah, let me check the chat window here. Okay, good. So like I said, I used this example to explain maybe shorter version of this, to explain the design principle of organ chip technology. And I hope this actually makes sense to you now, okay? Then, so this study was done, back in 2010. So kind of jumping in time, one of the questions, you could ask is what's happening now? What are the cool things, that are happening in organ chip field? So one of the main trends, recent trends in the field has been to develop disease models, because if you think about it, disease models are what drug developers need, right, to develop better drugs, and what clinicians and biologists, physiologists actually need to better understand how certain diseases develop, progress, and exacerbate. So this is developing or disease modeling using organ chip technology has been one of the main trends in the field. So what I'm going to do is I'm going to very quickly walk you through some of the disease models, organ chip-based disease models that we've been developing in my lab at Penn. Okay. So one of them is actually smoking lung on a chip. So I'm going to I'm not going to spend too much time talking about this model, but idea is, we have a very specific goal of studying what happens deep in the lung. So for the longest time, the distal part of the lung, deep in the lung, has been considered a silent zone. People didn't know much about what's happening in this, deep regions of the lung, but there's now a lot of evidence that this is where a lot of important and interesting things happen. So we are actually modeling this small area tissue. So the small area tissue, if you're new to this, consists of this columnar epithelium supported by this connective tissue containing blood vessels and connective tissue cells. So this is one example. of a device that we can use to model these multi-layered structures. All about, mimicking structure and environment first, and then, kind of they build themselves, kind of, okay? So to mimic this connective tissue, what we do is so this is a device consisting of 5 layers, 3 PDMS chambers separated by 2 porous membranes here. So we fill this middle chamber with collagen gel that mimics this connective tissue. And we also put in some fibroblasts, the cells that are found in the connective tissue in the lung. And then we culture epithelial cells under air in the upper chamber. We culture vascular cells in the lower chamber and flow media to mimic blood flow. So, as a result of cell differentiation, under air, at the air, so people call this ALI culture, air liquid interface culture. So lung cells become ciliated. They form cilia, hair-like structures on their top surface. They form tight junctions between them to form tight barrier tissue against, air and other harmful things in the airway system. And we also, came up with the way to put, keep these cells in this gel for a long time without any gel detachment or shrinkage. And so these are fibroblasts growing in this hydrogel. Over time, they start laying down their own ECM, in this case, fibronectin. The starting material is collagen, but we see fibronectin deposition over time. And we, use this system to deliver a cigarette smoke to the chip. And so we use an off-the-shelf cigarette smoking machine, generates, mainstream cigarette smoke, which is then humidified and diluted more than 96%, and it gets delivered to the smoking chip. I'll just skip all this, but the question is, what can we study? What types of biological responses and endpoints we can measure and analyze in this model system? So in one example, what we did was, just to skip this, to refresh your memory, this is what the stromal tissue looks like. That consists of the fibroblast cells embedded in type 1 collagen gel. Green shows a cell deposited fibronectin. So one experiment, what we did was we smoked those cells 1 cigarette a day, for 1 week, 2 weeks, and 1 month, see what happens. So we saw dramatic increase in the number of cells and also changes in the morphology of the cells. They become more polygonal, actually, which is actually, it's a typical morphology of a myofibroblast that are actually activated, and they produce a lot of ECM. And you can also see a lot, more robust staining of ECM around these cells. So this is actually airway fibrosis we're modeling, in this system. If you think about it, this is nothing new. Chronic smokers and lung cancer patients, commonly show thickened and stiffened airway walls because of this. But it was very interesting to be able to model this in this, man-made in vitro system. Again, I'm actually, omitting a lot of data, but we actually did studies to really validate the relevance of these results. And also in this paper, we're actually describing a study focused on using this platform to test adverse effects of electronic cigarettes. Okay. So before I move on, any questions about this?
Did you also just have normal cells growing in that system with like no smoke going in to see what would be the difference between the two?
Yeah. So we, for every endpoint, we had this pair of normal versus smoked, and we even tested the diseased actually tissues, and so they all show different responses. And so those changes, for example, cell stresses they experience, and also fibrotic responses they show, those were in comparison to normal tissues. But when we tested diseased cells actually, had a very different susceptibility actually to cigarette smoke. They were a lot more susceptible to cigarette smoke induced injuries. Let me just try to summarize, as that way. Yeah. Okay. All right. Okay, so this might be more interesting actually to many of you guys. I think it's freezing. Let me actually quickly apologize. Okay, let me restart my PowerPoint. Sorry about that. So the next thing I'm going to talk about is modeling cancer. So, and the treatment of cancer, I have to say. So this actually study was motivated by, so let me see, my screen. So we got this award from Cancer Research Institute about 3 years ago to develop a cancer immunotherapy on a chip. Let's see. So our goal was to develop, organ chip, a model of a vascularized tumor, and then use this system to study how cancer cells in the tumor microenvironment interact with circulating, immune cells, engineered to target, cancer cells. And so one of the first things we had to do was to develop refusable human blood vessels in microfluidic devices. So to achieve this goal, this is actually the device that we came up with. And this is a cutaway view of the device. And so the main design feature is are these two barriers protruding from the ceiling of the channel. So what we can do in this system is when we inject liquid into the center compartment, liquid actually nicely follows this middle lane. And when it gets to these barriers, it gets pinned along these barriers due to surface tension. So that's actually nicely demonstrated in this movie where we're injecting black food coloring. And due to pipetting pressure, there's a little bit of bulging, but it never ruptures and spills over to the side lanes. So using this technique, what we do is we inject hydrogel solution. We use a fibrinogen solution mixed with thrombin. So they, when they react with each other, you get fibrin, hydrogel. And we mix in fibroblast and vascular endothelial cells. So we form the gel in this manner. And then we fill these side chambers. We flow media through these side chambers to feed the cells embedded in the gel. And we also seed the side channels with endothelial cells. So we culture them in the gel in the side channels for about 4 to 5 days. And something amazing happens during this period. So the fibroblast cells embedded in the gel start secreting factors that induce endothelial cells self-assemble into 3D vessels that open to the side channels. So this is actually nicely demonstrated in this movie. If you look at this middle compartment, this is an overview of a channel. This middle compartment is where the endothelial cells expressing red fluorescent protein are residing. So over time they spread, but they also start connecting with one another to form 3D vascular structure. So this is actually a sped up movie. It takes about 4 to 5 days to form these beautiful vessels through the entire thickness of the hydrogel that we have. And it's really interesting to see these fibroblast cells wrap around those blood vessels as if they were parasites, right? And they have definitely open lumens. They have very small lumens. As you can tell, these are capillary-sized, micro vessels that we are engineering in this device. We've also developed a set of techniques that allow us to extract, these vascularized tissues from our devices after experiments. So these are tissue strips that we pull out from these devices, and which can then be, further processed for, further analysis. Like, we can take the samples to imaging court to get SEM images of, and this is a one blood vessel, and you can see also this, fibroblast kind of, in close proximity to this vessel. And this is a capillary, sized vessel that we were able to engineer in our system. We can also get TEM images. This is a TEM of one capillary in our device, which is very similar to in vivo vessel. And we're actually, mimicking the features down to the sub-micron levels. And so, oftentimes in vivo capillary vessels just show these cytoplasmic folds at cell-cell junctions. We see exactly the same thing in our microengineered in vitro vessels. And we also have the ability to tune the architecture depending on how many cells we put into the gel, how soft or how stiff the gel is. The CD31 staining shows endothelial cells, and these are all the same type of endothelial cells, but they show very different architecture, depending on these parameters. And these vessels are not very easy to quantify. Qualitatively, going back to this image, you can tell there's something different about these, different conditions, but it's really hard to quantify these the features. And so we're now using machine learning algorithm to skeletonize and also segment these blood vessels for 3D quantification. And what I'm showing in this image is, so there's an interesting thing that's happening at the interface between the hydrogel containing these red 3D vessels. And so green shows endothelial cells cultured in the side channels. So what's happening at this channel gel interface is that endothelial cells are forming sprouts into the gel, and as they go in, they run into these 3D vessels that are sitting in the gel. They actually connect with one another. So a fancy term for this is anastomosis, okay? So they anastomose with 3D vessels, and as a result, you get the open vascular network. So these guys, end up, or they actually open to the side channels through this process, and as a result, we can perfuse these blood vessels with a variety of things. In this case, we're perfusing the vessels with 75 kilodalton dextran, the fluorescent dye. We can also flow microparticles and nano carrier beads for targeted drug delivery. I hope the movies are playing smoothly on your end. We can also flow blood cells. We can flow RBCs on the left. These are T cells migrating around TNF, tumor necrosis factor alpha. This is a pro-inflammatory cytokine. Treated blood vessels, okay, to mimic inflamed blood vessels. Any questions about this platform so far?
Hi, Dan. I was wondering, how do you, what causes the Cubex cultured on the outer channels to migrate into this middle region and
So they,
anastomosis?
Yeah, anastomosis. Yeah. So it's built into their, their intrinsic programs. And so, when endothelial cells that sit on the surface of gel, they have a tendency to migrate into the gel. And if it's forming endothelial sprouts, it's a very common observation in 3D culture, 2D culture, hybrid culture systems. So if you think about, what happens in the body, you put something into the body, foreign object, it gets vascularized very quickly within a week, actually. You put a small piece of sponge, you take it out after 4 to 5 days, it becomes reddish, because of vascularization. So it's actually, it's in their program to form new, sprouts, new blood vessels to vascularize, objects, that they encounter. So, this process is called angiogenesis. And so what's happening at this interface is angiogenic sprouting of endothelial cells in the side channels. What's happening in the gel is a different process. It's called vasculogenesis. So this is kind of a de novo development of blood vessels during development in embryos. And so, you start with these, progenitorous endothelial cells, and they differentiate into endothelial cells, but at the same time, they kind of assemble themselves into 3D vessels. So that's actually self-assembly process driven by vasculogenesis. But, endothelial cells, angiogenesis is a built-in kind of intrinsic program in endothelial cells, depending on the environment they're subjected to. But in this case, we have VEGF in the media, so these cells are very angiogenic, I have to say.
Okay.
Okay.
Thank you. Yeah. And I was also wondering, I know this in the video of you perfusing the microbeads through the channel, that not all of the vessels are connected to each other.
Right.
Have you guys noticed any different factors that impact how, the degree of anastomosis in these devices?
Yeah, so it's heavily affected by the growth factors that we put into including the media and also gel properties, and so and also cell numbers, and whether or not you have perfusion or flow in the side channels. And so one of the interesting things we found out in our studies that if you flow media in the side channels, you don't get good anastomosis, you don't get good angiogenesis, because these endothelial cells secrete autocrine factors, paracrine factors, and these factors wash away when you have continuous flow in the side channels. So one of the new things that we do as a result of this realization is that we don't flow in the side channels. We just exchange media every other day to retain these factors. Okay.
Okay, thank you.
All right. Okay, so I've only covered 20% of my, what I initially planned on talking about, but so I'm going to fly through the rest of the slides very quickly. So, we are now using this as a platform. So, these are nice vessels, but, using these vessels, we can mimic, human tissues more realistically. So, for example, we can create vascularized islets. I can give another hour talk about this model, but, just know that we have this, ongoing study in the lab, using this platform to vascularize cadaveric islets, and we can maintain their function for long periods of time, in vitro. I'll just skip all this. We can also form fat tissue, white adipose tissue using a similar approach. So back to the cancer immunotherapy project. So this is a micrograph of a tumor explant that we got from our collaborator, Scott Worthen. We chopped it into small pieces, and we put small fragments into the device to vascularize. And as you can tell, they actually become vascularized, but they also grow, as a result of vascular supply in this device. And the vessels form in and around the tumors. The explants already have contained tumors, intrinsic and native, blood vessels. These blood vessels connect with anastomose with the engineered vessels surrounding the tumor constructs. And as a result, they become perfusable. So blood vessels perfusable. So, we can perfuse the tumors using these perfusable blood vessels. I'll just skip this. And so using this platform now, we are studying the immune cell, cancer cell interactions. Okay, so I'll just show you one example here. So in this example, we used lung cancer cells to form spheroids. We did not use tumor explants. We used tumor spheroids, but those cells were engineered to overexpress this tumor antigen called mesothelin. Okay. And we put CAR-Ts, chimeric antigen receptor T cell. These are one of the most commonly used engineered immune cells for immunotherapy. And so we actually put CAR-Ts engineered to recognize and engage mesothelin that I just talked about. So this is 2 hours after a cell infusion, and as you can tell, these CAR-Ts stick to the blood vessels associated with these tumors. And green cells are tumor cells. And within just a couple of days, some of these cells infiltrate into the tumors, and they're found in the tumors. And as a result of this, we see a dramatic effect actually. So this is without CARs, this is with CARs. So you can see a significant difference in terms of growth of these tumors. And so we saw significant tumor killing effects, or their effects on the growth of tumors. Again, so we have a lot of data, but I'm going to have to skip over some of these data, but we're actually using this platform to test the efficacy of different types of engineered immune cells, for the purpose of developing better immunotherapeutic, approaches, strategies. Okay. Let's see, 52. This is another example of airway disease model, lung disease model we're developing. So in this case, idea is the same. We culture epithelial cells, fibroblast cells, and vascular cells. But in this case, we're actually doing this to study asthma. So in asthma, when you have asthma attack, airway is actually narrow and constrict. So to model that, we use this hollow chamber to compress the chamber and squeeze the 3D tissue in the lateral direction to mimic bronchoconstriction. So we're actually using this platform. We've done a very nice study showing those pathophysiological mechanical forces during asthma attack, actually induce airway fibrosis in this model. So again, I'm going to have to skip over this in the interest of time. And this is probably one of the most exciting, interesting things we've ever done. So we're also sending organ chips up to space. So we got selected as one of the 5 teams in the country to send those tissue chips up to the station. So we're modeling immunosuppression. So astronauts get sick more easily during space flight, right after space flight, and there's evidence that the microgravity environment suppresses immune responses. And so we're modeling that using airway infection model hooked up to a bone marrow model. So the reason why we have bone marrow in this project is because bone marrow is the organ that produces all these immune cells, right? So we're modeling studying how this production and release of immune cells from the bone marrow into the vasculature changes actually due to microgravity. And we're actually linking these two systems to model the entire process from infection and then release of white blood cells into the vasculature, and then their travel to the infected lungs, and then their adhesion and recruitment transmigration into the infected airway tissues. And so, again, I'm happy to tell you more about this study offline. And we actually launched this device last year, and again, I'm happy to tell you more about these studies offline. Okay. So I have about 5 minutes left. So, so you can actually see one of these devices at MoMA. So the breathing law on a chip design actually was acquired into permanent collection in MoMA about 4 or 5 years ago. So I'm not sure if they still have this exhibit, but so here are the ideas, so far I've only talked about long on a chip model systems, but you can do the same thing for any other organs in the body, on a chip, right? So, and one of the kind of, well-known examples in my lab is eye model. So we're actually modeling the ocular surface of the eye, consisting of cornea at the center surrounded by the conjunctiva. So I'll just skip all this. But we were interested in mimicking eye blinking to mimic the environment. It's all about mimicking the structure and environment. Those are the first things we do. So this is the device that we came up with. It's called the Blinking Eye on a Chip, developed by CEMB, actually, student, former student, Jung-Yoon, in collaboration with 3 ophthalmologists at Penn. So this paper came out in Nature Medicine last year. So, if you're interested in this, feel free to, look it up and read more about it. This is actually what the device looks like. I'll just show you a movie and then move on. But we came up with ways to culture cells on this contact lens, shaped a scaffold. And we culture them under air to differentiate them. And this is the movie that shows the actuation of biomimetic hydrogel eyelid in this system. It slides back and forth to mimic blinking of human eyes. So, and in this study, we did a whole bunch of, things. And one of the interesting things that we did was in collaboration with Vivek Shenoy, we modeled biomechanical forces in this engineered system. To validate the relevance of this model. And also we found out that blinking forces actually help corneal cells differentiate in a more efficient manner, in a more rapid manner. So physiological blinking forces are good for the cells. That's actually one of the main findings of this study. And we also use this platform to model dry eye disease for drug testing. So again, in the interest of time, I'm gonna skip over this part. And there's one thing I wanted to briefly talk about before I finish. Sorry for the fly through. And just a quick mention, we're doing a lot of work in the area of reproductive biology and medicine. And so we're focusing on developing female reproductive organs like placenta on a chip and cervix on a chip. But anyway, so let me actually end by talking about what we think, the future is. So, organ chip technology, as you have seen, I hope I convince you that this is a great technology, interesting technology, but so it's great in that it allows us to mimic the complexity of human tissues and organs in ways that have not been possible. But the flip side is it's not very easy to make these devices. It's not very easy to work with these devices. It's very, actually, as a matter of fact, it's very challenging to make and work with these devices. So because of that, experimental throughput in typical organ chip devices is very low. So in our lab, for example, we used to be able to handle up to maybe 15 devices at a time. But if it goes over 15, we used to start struggling. So we took this problem really seriously, and we started developing higher throughput organ chip platforms and technologies. So we're trying to scale up by walking away from these, single channel containing devices. And we're actually beginning to use these devices, each containing more than tens of replicates. And this is the most advanced, platform we've ever built. This is actually a 6-inch wafer-sized device containing 128 individually addressable fluidic chambers controlled by very sophisticated, let's keep all this, pneumatic circuits. And so we use pneumatic pressure-driven flow valves to pump and valve fluid flows in this very complex, fluidic network. We've also spent a lot of time and effort to automate the manufacturing process. And so this device I just showed you, for example, has over 2,000 holes to punch, actually, to make access ports. And it takes about 4.5 hours to do this manually. So Andrei Georgescu is a graduate student leading this project, got really frustrated by this, and he built this hole punching robot from scratch. And so this is a robot in action. PDM is like butter. And between punches, the ejection pin, comes out to push out the core to prevent channel clogging with the core. And this is actually real-time movie of the hole punching. We can do this very quickly. We can go over, over 2,000 holes within 20 minutes. So we can go over a large number of devices very quickly, but also it significantly enhances quality of the hole punching. We have a very precise, spatial control. This is Andrei's pinky finger. And, we have multiple layers to bond and, align. So it's not easy to do that when you have more than 100 channels to bond and align. So he has also built a 2-point multi-layer aligner system guided by video microscopy, as you can see in this movie. We're also using building robots to automate fluid handling. So this is actually, for example, robotic gel loading into the blood vessel forming device that I just talked about. And we can also automate important steps of, cell culture, like exchanging media. So these, systems are making our lives so much easier, but more importantly, it's actually allowing us to drastically increase the amount of data that we can extract from these human cell-based organ chip systems. This has not been possible previously in typical organ chip, model systems. And so we're very excited about this technology, and we're in the process of commercializing this platform, out of my lab. And we also have a whole new area. Actually, we are venturing into organoids on a chip. And again, I don't have time to talk about this today, but we wrote a review for Science last year about organoids on a chip. So if you're interested, feel free to, again, that might be a good read. Okay, so with that, I'd like to end. And, if you have any remaining questions, I'll stick around and answer your questions. Okay, thank you for your attention.
Thank you.
Hi, Dan. This is Christine from Rowan University.
Hi.
And I was actually going to ask if it was possible to create a high throughput platform, and you've shown it, so that's cool. So
yeah.
I guess my question now is, do you see any application for this for creating screening platforms for personalized medicine?
Yeah, so personalized medicine is an area that we've all been very interested in for many years. To culture, the cells from patients, for example, one of the, major challenges has been low throughput, because, there's a lot of variability in those patient or, clinically derived materials. And so in low throughput systems, you would culture these cells and get results A, but next day you would do the same thing to get completely different results. And so with the help of higher throughput, culture platforms, developed in my lab, we're now starting to get a better sense of the variability, that is often observed in these, primary cells that we get from the clinic. So we do not have active ongoing studies to address those specific questions in personalized medicine, but we actually have begun to use patient-derived, cells, and we actually generated iPSCs from, for example, we have a retina model, and my collaborator actually has been collecting blood samples from AMD, age-related macular degeneration patients. And we've generated iPSCs from those blood samples, and then differentiated them into RPEs, retinal pigment epithelial cells, that we use to build a barrier model between the retina and blood vessels. And so using those patient-derived stem cells, we've been able to recapitulate some of those disease features. But the real question is, do you really see the patient-to-patient variability, and or maybe differential responses between these different patient populations? So we haven't gotten those results yet, but it's in the works. Yeah. And to tell you a little bit about the current use of this platform, so we're conducting SRAs, sponsored research agreement projects, actually 5 projects. It's surprising, that all these are happening all at the same time. But the GSKs, for example, very interested in using this platform to test, and for example, antivirals actually, against COVID-19. That's actually one of the studies we're conducting now. And cancer drugs, that they are developing. So they would like to use higher throughput platform, culture systems to test those drugs in a higher throughput manner. And GSK also has seen some very interesting side effects in human cornea, actually, as a result of administering this anti-cancer drug. And they like to see if we can recapitulate this, interesting corneal pathology in the blinking eye on a chip system. So those are some of the examples of kind of industrial partnerships that we have now.
Right. Very cool.
Yeah.
Thank you.
Yep. Thanks. Any other questions? So again, if you have any questions, comments, I'm available online all the time. So you can feel free to shoot me an email anytime. Okay. And I'm happy to talk to you more about some of those studies I briefly highlighted today. But yeah, feel free to do that.
Thank you very much, Dan. And to the students, so this concludes all the lectures for your boot camp. And so you have one last lab session later this afternoon. And so I will see you guys then. Thanks again, Dan. Great lecture.
Thanks guys. Yeah. Enjoy the rest of the camp. Thanks, guys.
Thank you.
Thank you.